Okay, as we continue to look at organelles, we notice we're still breaking them down in terms of their functional class that we've talked about, organelles that promote structural integrity. We talked about organelles that facilitate motility. Now we want to talk about a group of organelles that are involved in synthesis. Now this can be synthesis of proteins, this can be synthesis of lipids, a number of things, but we're going to kind of talk about how they do this. The synthesis of proteins at ribosomes, and then we'll talk about the difference between, well, we have this stuff called endoplasmic reticulum, and we've got one that's rough and one that's smooth, and then the one that was named after Dr. Golgi. So we'll talk about each of these and their role in the synthesis of molecules. Now the first one that's heavily involved in synthesis we discussed as we talked about protein synthesis, okay, ribosomes. And so these are little factories where we're going to assemble proteins, and we're going to put them together in the right order so that they then, of course, have the right function. So with our ribosomes, we have basically two parts that get together as one big functional organelle. So we have the large subunit, we have the smallest subunit. Now they have units associated with them. Okay, the S, so we have the 60S, the 40S. Okay, these stand for Svedberg units. Basically, it's a unit of, if we spin it in a centrifuge, kind of how fast it settles or how fast it moves. Well, obviously, a bigger molecule will move to the bottom faster than a smaller molecule. So those are the units that it basically involves. So a little bit of detail there, but nothing critically important to us. And so as we assemble these ribosomes, we assemble them into the complete ribosome that then allows us to put together these proteins. Now, these ribosomes can either be found as free ribosomes out in the cell, or they can be part of the structure of the endoplasmic reticulum. And because they're existing on that endoplasmic reticulum, it makes little bumps, and so we call it rough endoplasmic reticulum. Basically, the difference there is if this is a protein that's going to be used within the cell, then usually that protein is synthesized at a free ribosome. If it's something that's going to be packaged and shipped outside of the cell, then it's usually at the rough endoplasmic reticulum. So that's kind of the difference there of why we would have that. Now, with that endoplasmic reticulum, it's basically a network of membranes. And again, the rough ER, you hear it referred to as, or the smooth ER. The rough endoplasmic reticulum, the reason it's rough is because it appears that way because of the presence of ribosomes that are there. So we call it rough ER. So the job is, with the rough ER, is to produce large amounts of protein. So protein synthesis. Now the smooth endoplasmic reticulum, there's kind of three kinds of this. In a general cell, it's heavily involved in the production of lipid molecules. In some organs, and that's why we're going to discuss numbers 2 and 3 later, it plays a role in processing toxins and other cellular components like in the liver. And sometimes they'll have special names depending on their function within a given cell. And so, for example, in muscle, we have the sarcoplasmic reticulum. It's just a special name for smooth endoplasmic reticulum specific to that muscle's function. What happens in muscle in that sarcoplasmic reticulum, its version of the smooth ER, is it stores calcium. And when told, it releases that calcium to then get the muscular components to get together and allow a muscle contraction to take place. So it'll have a different name depending on where it's located, but it's still smooth endoplasmic reticulum. Now our next one is the Golgi complex. Okay? It's sometimes called, because of their structural appearance, Golgi bodies. So we have here the term Golgi bodies within cells, or the Golgi complex. It's the same, named after the Italian histologist, okay, Dr. Golgi. Kind of the joke that's kind of made is it's the shape of his mustache. We'll notice the shape of the Golgi complex. So these are kind of like floppy pancakes, and they're stacked. And what's basically happening here is that we have produced these vesicles, okay? And they're coming from the rough endoplasmic reticulum. So what do they contain? They contain proteins. So we have vesicles containing proteins that enter into the Golgi complex, and they'll pass through those layers of membranes and exit out as secretory vesicles on the other side. And that's why I mentioned earlier that proteins constructed at free ribosomes tend to stay as intracellular proteins. Proteins constructed at the rough endoplasmic reticulum tend to be then sent extracellularly because they get packaged, and so they're allowed to do that through vesicular transport. So here's a graphic that contains a little bit more detail on what's taking place here. And I think maybe the easiest way to think about this, it's kind of like, okay, you need to mail a package. This package, let's say it's going to go internationally. And so you take it into the UPS store. Okay? You take it into the post office, but let's use the UPS store as our example. You take it into the UPS store, and the attendant there looks at it and goes, yeah, the luck of that getting there is so not happening. Now, sometimes that can be simple, that all they do is relabel it. Sometimes that can be complex, that they're like, yeah, you know, to not spend so much money, let's repackage it this way, or to keep it from being damaged, let's repackage it this way. So the function of the Golgi complex is to repackage and modify and change these things so that they're ready to be sent where they're designed to be sent to, okay, and to have the function that they need. So that's what's basically happening here, is that protein is produced, it's released from the rough endoplasmic reticulum in a transport vesicle. It then enters, okay, what we call the cis phase. So there's two phases on this. There's the entry or the cis phase, and there's the exit or the trans phase. And so it enters the cis phase, passes through the layers of membranes, is modified, clipped, tweaked, things are added to it, things are taken away from it. It then, through a secretory vesicle, is then shipped out the other side. So these can be proteins that are going to merge with the cell membrane, like we talked about previously. These can also be things that are exported outside the cell. So either way, but they go through kind of a repackaging process in that scenario. Now, our next group of organelles are involved in storage and digestion. And we introduced these a little bit when we talked about phagocytosis, pinocytosis as forms of endocytosis, that we involved, for example, the lysosome. Well, we're going to add to that, we're going to add two more. We're going to add a peroxisome, and we're going to talk about a proteasome as well. So the lysosomes are actually formed as vesicles from the Golgi complex, and their job is to digest and allow things to be recycled or allow things to be destroyed and disposed of. So in a worst case scenario, if the cell is sick or dying, the cell actually has the ability to kind of flip a cell suicide switch, where it's like, all right, I'm going to take one for the team, such that if the conditions arise that things aren't very conducive to that cell doing very well, it can flip a switch and release the contents of its lysosomes and actually destroy itself. So through an autolytic process, lysis means to break, auto, self. So through a kind of autolytic process can implode itself, and then portions of that cell can then be recycled. Now, the way it facilitates this is these enzymes, which are very powerful, work best at an acidic pH. But that's okay because we are protected because of the membrane around that lysosome, and it's a phospholipid membrane. And then the digestive enzymes are contained inside. Enzymes specific to digestion of proteins, enzymes specific to digestion of lipids, enzymes specific to whatever it is that is involved in that cell. And so by the use of hydrogen ion pumps, it facilitates that the pH in that lysosome is very, very low. Now, as we look at our next organelle involved in digestion, it's called the peroxisome. So they're like lysosomes, they're membrane bound, and they destroy materials for the cell. They contain enzymes, and specifically a higher concentration of enzymes called oxidases. And so as they target molecules, the product of those is going to make hydrogen peroxide, H2O2. Now, H2O2 or hydrogen peroxide by itself can be toxic and is toxic to your cells. So we have other enzymes that break down hydrogen peroxide into, we break it off, and one of the products is water. So it lessens the impact on our cells. So these are common in the liver and the kidney because of their detoxification functions. They also help us break down fatty acids for use of energy through the process of of beta oxidation. But that's the peroxisome, a little more specific in what they do. A part of what they do is they do produce free radicals. So in a previous discussion, we talked about how through oxidative reactions, we can produce free radicals. So that's why to lessen the production of free radicals, we utilize molecules, both endogenous and exogenous, that are antioxidants to lessen the amount of free radicals that are produced. Now, the last one of the three, the proteasomes. Now, the job of the proteasome is to basically get rid of foreign, screwed up, misfolded, misconstructed proteins. And so these might have been proteins that only needed to be used temporarily. These might have been proteins that are coming from microbial invaders. These may be proteins that just didn't get made right. And what happens is that misfolded, misguided, or foreign protein will first be tagged by a protein called ubiquitin. And so that's how the proteasome knows, okay, which one do I need to tear apart? Well, it gets flagged and told which one needs to get torn apart. And so what happens is the proteasome then breaks apart that protein, and then we can recycle those amino acids. And so that's the function of the proteasome.